Good afternoon. Thank you for the kind words. And I would first like to thank the organizers for giving me this opportunity to talk about some of our work to you here. And also let me say that I'm absolutely delighted to be in Wageningen in person for the first time. It's a beautiful place, and I'm really enjoying the visit and enjoying the hospitality of the hosts. So let me begin by quoting a good friend and scientist, Dr. Isaac Cohen. Medicine is too important to be left to the doctors. So what does he mean? What does Zach mean by this? So I paraphrase here. What he means is that the doctors all around the world are doing a good job of keeping us in good health, given the state of the knowledge. But the boundary of this knowledge can be pushed even further by using bioinformatics tools on data that already exists in hospitals and in healthcare systems. Okay? And bioinformatics did not even exist 20 years ago. It was not a subject, it was not a discipline, it did not exist. Now bioinformatics is ubiquitous, and it is almost indispensable for every biological research. Every biologist, sometime or the other, uses bioinformatics tools. But bioinformatics has been nurtured by biology. So in the next 25 minutes or so, I want to explore this symbiotic relationship that exists between computer science and biology. But of course, compared to biology, computer science is a mere juvenile. Yet, biology has nurtured and fostered bioinformatics into becoming a self-respecting discipline. And I'll also talk to you a little bit about what is happening in computer science, that is the cognitive era in computer science. And then in the second part of my talk, I want to give you two reports from the trenches, so to say, on projects from my group, from IBM Research, one on cognitive genomics or precision medicine, and the other is on big data genomics or big data metagenomics. As Professor Arthur just mentioned, at the turn of the century, biology saw a landmark event when the human genome was sequenced and the assembly was completed. It was a remarkable event. It was brought about by advances in biotechnology, which made it possible to read single base pairs from fragments of DNA. And the other was clever algorithms, or bioinformatics, which had the ability to take a bag of millions or 10 million fragments and put them into one single coherent sequence of the genome, which is 3 billion bases. It was remarkable even for algorithms, and this was the first time that algorithms played such a vital role in a project so ingrained in biology. But of course, we underestimated the variations that exist within humans, that is, within the same species. So we have projects to actually capture the variations that exist across humans. And we are sequencing animals, the 1,000 Bull project, backed by the meat industry, and the 10K genome project, which aims to sequence 10,000 distinct animal sequences. And with an eye on extinction, understanding extinction, probably preventing extinction if possible. Of course, plants are not left behind. There is the 1001 Arabidopsis project. And I mentioned cacao, not just because I love chocolate, which I actually do, but also because my team was involved in the sequencing effort of the cacao genome. But of course, nature plays catch me if you can with us, scientists. And we again had underestimated how much effort it takes in understanding the data that we are gathering. So the genomic data that we collect, we need to understand it, and so now we have in the process of trying to understand, we have to gather more data, which is phenotype data, transcriptomic data, and related data. And the mother of all phenotype projects, the Brain Initiative, which was announced a couple of years ago, and there is this endeavor to map the brain. Bioinformatics is doing a good role here. It's based on string algorithms, based on graph theory, based on combinatorics and statistics. But the data sizes are exploding. As Professor Arthur said, it's increasing in size, modality, and so on. So what I'm going to do now is I will switch lanes slightly and go into computer science. Okay? And at the risk of oversimplifying, computer science problems, there are two kinds of problems there. One is on factoring very large numbers or finding the most optimal route for a traveling salesman. And on the other hand, computer scientists are fascinated by toddlers. How does a toddler avoid obstacles? How does a toddler recognize objects, recognize faces? How does a toddler learn languages? Okay? And that has given rise to the field of artificial intelligence, or AI, in contrast to human intelligence or natural intelligence. AI has been around since the 1980s at least. But it saw its own watershed moment in 2011 when a program that was designed and developed in IBM called Watson defeated the world champions of Jeopardy. And the media called it the smartest machine on earth, and arguably so. For those of you who are not familiar with Jeopardy, Jeopardy is a game based on general knowledge or trivia. It is extremely popular in the US. It has an extremely large following. It is telecast almost every weeknight in multiple channels. And it has large prize money associated with it. And like professional sports, it attracts the best talent. So it was indeed remarkable that a program could beat the two champions who were actually unbeaten for many rounds. It was able to beat the two human players on national television. And that has been dubbed as the dawn of the cognitive era. And in the short 5 years from Jeopardy, now Watson has made inroads in various industries, like the financial industry, in security, in call centers, healthcare. So how does Watson do it? To understand that, I will introduce you to something called the Watson Discovery Advisor. It's like an assistant. It's an avatar of Watson. It's a research assistant who sits by your elbow and helps you in your research. So let's watch a video on this.
Breakthroughs in medicine come from many years of hard work and some serendipity. With 21,000 genes, 193,000 clinical trials, 7,000 drugs, and 1.8 million articles published annually, science is saturated with data. But can we harness that data to help enhance innovation and accelerate discovery? IBM's Watson Discovery Advisor can help researchers make connections that may not have been previously considered, which may lead researchers to new insights and breakthroughs. Let's imagine a researcher and her work investigating the role of the LCK gene in ovarian cancer. As a kinase, this could be a new drug target, but more research needs to be done to understand the role of LCK and its relationship to other genes, diseases, and drugs. Unlike keyword search tools, Watson understands the language of cancer research. When the researcher searches LCK and ovarian cancer, Watson looks for all of the aliases and synonyms for that gene, disease, or compound of interest. Watson understands the nature of relationships between entities by recognizing verbs, pronouns, and other key parts of language. Watson evaluates the millions of pages of scientific journals, patents, chemical data, and other data, looking for any direct connections between LCK and ovarian cancer. In this case, the only relevant passage Watson found discusses a drug that increased LCK expression, but did not produce a positive result in ovarian cancer. On the other hand, Watson did identify several top genes, which the researcher wants to explore further in the biological entity network. She will explore whether there is an indirect relationship between LCK and ovarian cancer through other genes. Using a foundation of machine learning and domain-specific training, Watson recognizes new genes, diseases, and drug entities and can...
So let me stop here. And I hope you got an idea of how Watson learns or how Watson is making inroads in different areas. Of course, the key takeaway from here is domain-specific training. As they say, the mantra is context, context, context. So the context here was cancer research, and I will now switch to my first report from the trenches, so to say, on personalized oncology. So the context is similar but different. And we call this Watson genomics, and our focus is on cancer because cancer is believed to be a disease of the genome, so hence a good candidate for Watson genomics. So what does recent literature tell us about personalized oncology? It says that molecularly targeted therapies for patients, which is based on the molecular profile of the patient, is actually giving good results. It is resulting in improved survivability of the patients, if not in all, at least in some. But this cutting-edge treatment is available only in certain cancer research centers, and most patients have to travel far and wide for this. So what will it take? What will it take for us and the clinicians to bring this opportunity or this possibility of an exceptional response to every single patient? So we need a system that is objective, so that is not biased by the geography or biased by the cancer center. We want it to be fast, turnaround time should be faster. We want it to be transparent. There is no room for black boxes here because we need the rationale and the justification for what Watson would tell us. We want it to scale, of course. We're not talking about tens of patients or hundreds of patients, but maybe thousands or even millions. And such a number would mean that you need to computerize or automate the system. And finally, we want the system to be comprehensive. We want it to reflect the current state of knowledge in that field, and we want it to be cognizant of all the research articles that exist in this domain. So this is the sine qua non of this system. And I will spend some time trying to explain how we deal with this aspect of the system. So to put it in context, let's see the etiology of cancer. As cells divide in the tissues in our bodies, they undergo mutations. Most of the mutations are harmless, but some of them are not. And these harmful mutations begin to accumulate over a period of time, and the phenotype of the cell changes, that it goes from a normal or a precursor cancer cell to benign, to malignant, to metastatic. And how is it useful for us? Because this is the footprint it leaves on the molecular profile of the patient that our system can go and look at, or any such system can look at. Here we need to understand another concept, which is called the driver gene versus passenger genes. So just like if you want to stop a bus, you must target the driver. It is not very effective to target the passengers to stop the bus. Similarly, there are genes which are driver genes, and your therapies must target these driver genes. So in the personalized oncology context, it is important for us to find these driver genes. But unlike a bus where you know who the passengers are and who the driver or drivers are, it is not so clear in the molecular profile of a patient. But that's a challenge we have to face. There is another notion we need to know, which is that of oncogene or tumor suppressor gene. Oncogene is often called the accelerator of the car, and the tumor suppressor gene is often called the brake of the car. And this is a different kind of footprint that is left by the patient when we look at the gene expression. So overexpression is associated with oncogenes and underexpression or inactivation with tumor suppressor gene. And this is something also that one can exploit when one is looking at the molecular profile of a patient. And the molecular profile, or the NGS data, next generation sequencing data, composes of the genomic data, the epigenomic data, the transcriptomic or the expression data, proteomic data, and such similar data set. We are currently studying epigenomics and the proteomics, but the genomic and transcriptomic data is something that we are already testing it out with real users and doctors. The other point I want you to note is that in this case, even for a single patient, the data size is extremely large, just for one patient. And it is in terms of millions to tens of millions of pieces of information. It is not bits or bytes. It's not ones or zeros. It's not bits or bytes, but it is pieces of information that is potentially usable for that patient. And this is just for a single patient. There are other difficulties which makes it difficult to reach out to off-the-shelf solutions that exist in machine learning or data mining. Some of these difficulties are that there is rampant heterogeneity. Of course, the disease varies from patient to patient. That's why it makes it possible to do personalized oncology. But it makes it difficult to understand the disease. The portions of the tissue within the same patient also varies based on which part of the tissue, which part of the organ it was taken. And the forces that fuel a driver gene is through different mechanisms like SNPs, single nucleotide polymorphisms, or copy number variations. And they vary from patient to patient for the same disease. So there is rampant heterogeneity. But in the same breath, I say that there is rampant homogeneity. When the data is viewed differently, you begin to see similarity of behavior through co-expressions of genes even for different diseases and different patients. The other difficulty is that our knowledge of the disease is incomplete. Our knowledge of cancer is, of course, incomplete. We think, the research community, that the common variants have perhaps been completely cataloged, more or less, but the rarer variants, which are not common, we still discover them as we go. And when the problem is viewed as a prediction problem, it is acutely underspecified or undetermined, say for a typical machine learning approach. That is, the number of features are in millions to tens of millions, but the number of patients are in thousands, even when you include the cell lines. So as a result, there are a lot of difficulties in, may I say, predicting the driver genes given the molecular profile of a patient. So we have devised our models and algorithms to deal with this push and pull of different views of, in the area. And if there are any gaps there, we fill the gaps by engaging Watson to give us information. Just like a mathematician has a different way of reading, understanding his or her mathematical research papers, which will be different, say, from a chemist, the way he reads, understands, and internalizes research papers in chemistry, so must Watson be trained to read specialized literature in this. So as you saw in the video, we first build the dictionary, so it needs to know what are the words that are relevant in this field, the groups of the words, what are the different categories of words, and what are the relationship between this. But it needs to be trained so it can actually ingest and be useful to our system called Watson Genomics. So to summarize, the way Watson works, or Watson Genomics works, is it takes the patient's molecular data. It has ingested 20 plus million articles, research articles. It uses information it has gathered from them. We also look at some structured data sources. Structured data sources are those where the community has cataloged the knowledge in terms of what SNP causes, what disease, what it is most associated with, and so on. So it has 20 plus structured resources and 23 million articles, and it uses its internal model to give a ranked list of driver genes, which it thinks as driver genes for the patient, along with drugs that can be targeting some of these driver genes. And this is deployed in the cloud in a multi-tenant as a multi-tenant web service, and there is appropriate security, industry standard security safeguard is also implemented with this. So the output that Watson provides is for the molecular pathologist to see what are the driver genes, what is possibly a pathway. So this is what Watson is hypothesizing as the cause of the disease of that patient, as well as for the clinical oncologist in terms of the list of the driver genes and what are the drugs that they could be targeting. So if I had not clarified earlier, the idea is not to replace the molecular pathologist or the clinical oncologist, but to help them take decision by providing them solutions and also providing them layers of information in terms of why Watson comes up with these hypotheses. There are many metrics by which you can measure Watson genomics. What is on the top of our list is accuracy. Accuracy in terms of the driver genes, accuracy in terms of the drugs. So there are two measures that we use, which is precision and recall. Precision measures how correct is Watson's answer, how correct are the genes that Watson comes up with, and recall is what did Watson miss. And as we work with the clinicians and their end users and get their feedback, Watson is constantly learning and it is improving with time. Very happy to say we have many early adopters, which includes top cancer institutes and hospitals in the US. And there are many more talking to us as we go. And we have received good endorsements from them. And of course, Watson is learning. This is a learning process, learning for us. We are constantly trying to bring in more functionalities to Watson genomics, but we work very closely with hospitals and with cancer research centers, with clinicians and pathologists on this. So what are the future challenges? I call them more from less and more from more, and both driven by biotechnology. Don't be alarmed to see ctDNA, that it is not chloroplast DNA, it is circulating tumor DNA. They seem to have the same acronyms in both the fields. So the first biotechnology is driven towards non-invasive techniques. Like instead of getting a biopsy of your tissue, one could just prick your finger, get a drop of blood, and use that to retrieve the molecular profile of the patient. Of course, here, your analytics and Watson has less amount of data, less modalities of data, yet you want it to give as much correct and accurate result as possible given this less information. That's why I say more from less. And this is a challenge for analytics and for Watson. And the other is more from more, which is single-cell genomics, single-cell cancer genomics. You may or may not have realized is when we are talking about the molecular profile, although it corresponds to the molecules that exist in a cell, but the profile that we get is an aggregate of many cells. While single-cell genomics can give you the data on a cell-by-cell basis. This is going to lead to further explosion in data. Of course, there is more information, which is not averaged out from the other aggregate. But your analytics has to cope with the other explosion that you're having. And just to get an estimate of how many cells are there in a human body. I quote a research paper here, of course. It is estimated that there are 37 trillion cells in an average 30-year-old male who happened to be European of average height, 0.58 or so, but 37 trillion cells. But of course, for single-cell cancer genomics, we don't have to look at all these 37 trillion cells, but when we start doing single-cell genomics, this is the kind of number that we talk about. And that is also a challenge that we have to take up in the future, which is handling this data, which is more from more. And it is estimated that the number of microbes we carry in our body is 3 to 5 times the number of cells in our body, which is 100 trillion cells. And that brings me to the next report from the trenches, which is on big data metagenomics. Microbes are everywhere. They are in the air, they're on the surfaces, they're on our bodies, and they're playing a good role. And with time, we are getting to understand better how microbes work. And even the popular media talks about it. So here is a picture that is worth 1,000 words. And I really like the title. Some of my best friends are germs. And they help us, and they also play an important role. It has been seen that genetically identical mice, the experiments with genetically identical mice sometimes are not reproducible in different labs, perhaps because of the change in environment and change in microbes that they carry. And the microbes that we as humans carry, the ones that we developed since we were born, after we were exposed to our mother's microbe, is so important that doctors are contemplating swabbing babies which were not exposed to this, that is, they were born through cesarean section or C-section. They're contemplating swabbing them with their mother's vaginal fluid. This is how important we think that the microbes that we nurture since our birth is important. And the microbes in our gut keep us healthy, but they've also been implicated in malnutrition, that is, composition of certain microbes. Constellation of some microbes cause malnutrition. And then there are some of these bad guys that we hear about. Just yesterday, I was reading about this article that Starbucks is plagued now with this listeria problem. And there is Salmonella, Campylobacter, norovirus, and so on. These cause illnesses. 1 in every 6 individuals is going to be affected by one of these food-borne illnesses. And they sometimes cause death. And death in thousands, even in the US. And they come at a big cost to the economy. The US spends $3 to $4 billion a year towards salmonella infection-related incidents. So IBM and Mars are launching a food safety consortium to understand these microbes, or the constellation of microbes that exist, that reside in the food supply chain, which now is global. So this is a consortium that is trying to understand this to reduce the kind of numbers I just mentioned. And this has been heralded as one of the largest such efforts in a metagenomic study, with terabytes of data. And I'm sure this is a very conservative estimate of the number here. And what IBM brings to the table is big data and the accessories that go with big data, the platforms that will go with it, and, like I said before, context, context, context. So we also come with context-specific, domain-specific analytics, which we have built over the years, say working on, say, plant genetics. We have worked closely with plant geneticists over the years, building analytics. And I'm also proud to say that we have used this cutting-edge technique, a mathematical technique called topological data analysis, or TDA, on microbiomes. What TDA does is that it helps you see hidden structure in very high-dimensional and noisy data, which is microbiomes for us in this case. And the problem we were looking at was host prediction based on the microbiome composition. So to conclude, I come back to the theme of today, which is digitalization of nature. So how do we go from digitalization of nature, collecting data, all the way to knowledge and wisdom? Bioinformatics has put us in the right path, but I hope I've made a case, I've convinced you. And you probably already are convinced that we need this arsenal from computer science, which is big data, cognitive computing, analytics, the context, analytics, and a slew of cutting-edge techniques like TDA that exist which should propel us towards the understanding that we finally want. So, if I may, I want to end with this statement. Thank you.